speakers of today's event, Dr. S.C. Lakotia, Distinguished Professor, Department of Geology, Banaras Hindu University, Varanasi, and Dr. L.S. Seshidhara, Professor, Biology, and Director, National Center for Biological Sciences, Bengaluru, and Dr. A.K. Singh, Director and Vice Chancellor, IRI, and Dr. Gopalakrishnan, Head Division of Genetics, IRI. Ladies and gentlemen, please put your hands together to extend a very warm welcome to the dignitaries on the dais. Thank you. So now let us proceed with seeking the blessings of the Goddess of Knowledge, Ma Saraswati. I request the graduate school students to render Saraswati Vandana, a prayer to seek wisdom, knowledge, and guidance for the event ahead.
जय हे भगवती सुर भारती तव चरणों प्रणमाम्यहम्। जय हे भगवती सुर भारती तव चरणों प्रणमाम्यहम्। नाद ब्रह्ममयी जय बागेश्वरी, नाद ब्रह्ममयी जय बागेश्वरी, शरणं ते गच्छाम्यहम्। जय हे भगवती सुर भारती तव चरणों प्रणमाम्यहम्। शरण्या त्रिभुवन धन्या, त्वमसि शरण्या त्रिभुवन धन्या, सुरमुनिवन्दित चरणा। नवरस मधुरा कविता मुखरा, नवरस मधुरा कविता मुखरा, स्मित रुचि रुचिरा भरना। जय हे भगवती सुर भारती तव चरणों प्रणमाम्यहम्। हंसे आसीना भव मानस हंसे, कुंद तुइन शशि धवले। हर जनतां कुरु बोधि विकासम्, हर जनतां कुरु बोधि विकासम्। सित पंकज तनु विमले, जय हे भगवती सुर भारती तव चरणों प्रणमाम्यहम्। प्रणमाम्यहम् 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 प्रणमाम्यहम्।
Thank you, students, for that beautiful invocation. As we embark on the journey of Golden Jubilee celebrations of discovery of wingless mutant of Drosophila, let's symbolize the dispelling of darkness with the lighting of lamp. I request the dignitaries on the dais to kindly come forward and do the honor of lighting the ceremonial lamp. दीपम ज्योति परब्रह्मा दीपम ज्योति जनार्दना दीपो में हरातु पापम दीपम ज्योति नमोस्तुते. I fold my hands before the light that brings prosperity, auspiciousness, good health, abundance of wealth, and destruction of the enemy's intellect. Thank you. May now request our beloved director, Dr. A.K. Singh, to extend a very warm welcome to the dignitaries with a bouquet of flowers. To the chairman, Professor R.P. Singh. To the guest of honor, Dr. Trilochan Mahapatra, who is joining us online. Thank you, sir. I again request you to kindly deliver the formal welcome address and also introduce the chairman and guest of honor to the audience.
A very good morning to each one of you. Padma Bhushan Professor R.B. Singh, former Director IRI and Chair of today's function. Professor R.P. Sharma, former Director, National Research Center on Plant Biotechnology and President, 15th Genetics Congress Trust and Chief Guest of today's function. Dr. Trilochan Mahapatra, Chairperson, Protection of Plant Varieties and Farmers' Rights Authority, and Guest of Honor for today's function. Dr. Mahapatra has joined us online. Learned Speaker of the Day, Professor S.C. Lakotia, Distinguished Professor, Department of Geology, Banaras Hindu University, and Professor L.S. Sasidhara, Director, National Center for Biological Sciences, Bangalore. My colleague, Dr. Gopala Krishnan, Head Division of Genetics, scientists, students, participants who have joined online, ladies and gentlemen. I extend a warm welcome to each one of you to this very important function today when we are celebrating Golden Jubilee of discovery of wingless mutant in Drosophila melanogaster. Professor R.P. Sharma identified the first wingless mutation in Drosophila through EMS-induced mutagenesis at the Division of Genetics IRI and reported it in Drosophila Information Service in 1973 and subsequently published full-length paper along with Professor V.L. Chopra in Developmental Biology. The gene wingless is the first member of Wind gene family. Mutations in the Wind gene lead to a specific development of defects and their perturbation are linked to cancers in humans, including colorectal and skin cancers. Following the footsteps of Professor R.P. Sharma, Dr. Shanti Chandrasekaran continued working on Wind gene, namely DWINT4, 6, and 10, along with Wingless. Her untimely passing away was a huge loss to Drosophila genetics. Now Dr. Prachi is continuing their great legacy. While celebrating Golden Jubilee of discovery of wingless mutation in Drosophila, we are extremely happy to have among us the father of wingless mutant, Professor R.P. Sharma. Equally pleased we are to have with us two leading Drosophila geneticists of the country, Professor Lakhotia and Professor Sasidhar, as a speaker for the day. Once again, I welcome you all, sir, to this very important function. This program is being chaired by Professor R.B. Singh, former director of IRI, and he has been part and parcel of IRI all along. Professor Singh did his bachelor and master's from Patthar College, that is C.S. Azad University of Agriculture and Technology, Kanpur, PhD from North Carolina, served at Punjab Agriculture University, CCS University Meerut, Himachal Pradesh Agriculture University, and a long stint at Banaras Hindu University. Leading geneticist, he contributed significantly to development of large number of wheat varieties and varieties of pulses. And then he joined at the FAO as Regional Plant Protection and Production Officer. After serving a long stint at FAO, he came back to serve motherland as Director of IRI and as Chairman SRB. Again, he went back to FAO as ADG. And on coming back, he served as member of Farmers Commission and also as President of National Academy of Agricultural Sciences. He has made tremendous contribution to national and international agriculture, and recognizing his contribution, Government of India conferred him with Padma Bhushan. Thank you, sir, for accepting our invitation to be chair of this important function. We are joined by Dr. T. Mahapatra, former Director General ICR and Secretary there, and currently Chairperson, Protection of Plant Varieties and Farmers' Rights Authority. As a matter of fact, it was the initiative and idea of Dr. Mahapatra that when we are completing 50 years of the discovery of wingless mutation in Drosophila, we must have a celebration, and that's how we are here today. Because of some commitment, he could not be here with us, but he has joined us online, and I would like to extend a warm welcome to you, sir. Dr. Mahapatra, as you all know, has been a distinguished teacher, a celebrated teacher of this institute, who has been associated with teaching for more than 20 years at this institute, and his classes used to be full of students. In this hall, we find a lot of seats vacant. We are expecting a lot of students to be here, and hopefully they will join soon. Dr. Mahapatra classes always fell short of chairs in the classroom. That was his popularity as a teacher. He has made significant contribution to genome sequencing of varied crops, including rice, tomato, and many other crops, and to genome mapping, molecular breeding in rice. And as a research manager, he has brought a large number of reforms in the Indian Council of Agriculture Research. We are indeed very grateful to you, sir, for joining online and being with us. And I once again extend a warm welcome to you. Thank you very much.
Thank you, sir. May now request our guest of honor, Professor Trilochan Mahapatra, Chairman PPV FRA, for his opening remarks. Sir,
very good morning, and thank you very much. Thanks to all of you for being there. And respected Dr. R.B. Singh, Chairman of the program, Professor R.P. Sharma, Professor Lakhotia, Professor Sasidhar, Dr. A.K. Singh, Dr. Gopal, all on the dais, and everybody off the dais who are present in the hall. it's a pleasant day and most memorable day that we are celebrating 50 years of Wingless Mutant developed by Professor R.P. Sharma. In fact, when I was Director General in 2022 December, I was scanning through the discovery and trying to find out how many years it is. Then I noticed that 2023 would be 50th year, the golden jubilee year of the mutant. And then, in the meantime, because I left the position and then forgot about it, and suddenly discovered that again the time is running short and we should have this program. And that is how, I thought probably we should have this particular one day or half a day celebration with lectures to learn from experts to enrich our understanding in the field of Drosophila in general and Wingless in particular. And we cannot have, better speakers than Professor Lakhotia and Professor Sashidhar to enlighten us today in the arena of Drosophila genetics and also very specifically the Wingless and Wnt signaling pathway, and particularly the Wnt being homologue of Wingless, Drosophila Wingless, and its involvement in particularly cancer, and how at the membrane level the Wnt product behaves and how it operates very specifically and brings about changes and so on and so forth. And we would be certainly learning more from the speakers. But very briefly, I have been privileged to be a student of Professor R.P. Sharma, learning from him the fundamentals of molecular genetics. He has been an involved teacher and exposing and telling us those basics using Drosophila, the Mendelian laws, the law of segregation, law of independent assortment, and other even interactions, and using Drosophila model and demonstrating those through practicals, through theory, and modern molecular genetics principles, whether it is RNA splicing or even protein level modification. So through his molecular genetics course, we learned so much, and the foundation that he has laid, we all immensely benefited from that during those days when he was teaching and taking those classes. This is one, and which is at IRI, at the Biotech Center subsequently of IRI, and now National Institute of Plant Biotechnology, where he served as a director and guiding a whole lot of new areas, particularly the development of transgenics in case of crops. And that is what he pioneered in the country. And transgenic brinjal is the one which was developed fast in the country. That was, anybody else venturing into it, Professor Sharma initiated that. And then subsequently, when, after 2000, after Rice genome, when we were discussing functional genomics, that how to really move from here, and genomic sequence and what to really do, and he is the person again introduced mutagenesis, what he carried out in 50s and 40s, and the 60s rather, 1960s. Then he's proposed, and rather he led and guided us to develop EMS-induced mutations in rice. And the product of that is the herbicide-tolerant rice today, and varieties which have gone to field now. So again, another important milestone in his research career of mutagenesis, that he started in Drosophila, and taking it to the stage of application in a crop plant like rice in the form of herbicide-tolerant mutant. the mutant which was induced in the background of a variety called traditional variety called Nagina 22. That gene has gone to a very good background by Dr. A.K. Singh and his colleagues in Basmati ones, including Pusa Basmati 1121, which is exported, and we earn more than 15,000 crore annually. So this is what Professor Sharma, who has been even after his retirement, 20 years after his retirement, he used to come to field and get involved in selecting those mutants and standing in the field for hours together. After 20 years of his retirement. Now that is the involvement Professor Sharma has and continues to have and guiding the researchers in the realm of genetics and molecular biology as a whole. And we all express our sincere gratitude to you, sir, for being a pillar of strength and guiding generations of researchers at IRI and subsequently at NIPB. And I being the beneficiary, I express my sincere gratitude to you. And thank to Dr. A.K. Singh for having this program. First of all, to enrich everyone's knowledge in Drosophila, and Drosophila work still is continued at IRI for the benefit of students particularly, and very importantly also to felicitate Professor Sharma for his monumental contribution. Though the work he published in developmental biology could not be continued, as it is being done elsewhere, by him, because he branched out to many other things. And rice genome work, in fact, under his guidance, rice genome sequencing was actually initiated when he was project director of National Institute of Plant Biotechnology. So thanks to Dr. A.K. Singh for taking this initiative and for the benefit of everybody. And thanks to Gopal, head the genetics for organizing this, and everybody else. So with these words, I express my sincere gratitude to both the speakers of the day and Honorable Chairman, Professor R.B. Singh, for agreeing to chair the particular program, and very specifically Professor Lakhotia and Professor Sasidhar, for sparing their valuable time. And I'm deeply, honored and, in fact, grateful to both of you for agreeing to be part of this program. And thank you very much. And thanks to everybody who have spared their time to be present in the program. I am sorry I am not physically there, so I'm here at NBRI Lucknow for their Research Council meeting. So I'll be missing the deliberations because I have to be here, attend the proceedings here. So, but I wish the program a grand success. Thank you very much, and beg pardon for not being there and not being able to attend the whole program. Thank you very much. So, namaskar.
Thank you, sir, for setting the tone for this event with your thoughtful words. Your wisdom and perspectives have truly enriched our gathering. So may now request our ever-young and dynamic Professor R.B. Singh, who is chairing today's event, to do the honor of introducing the chief guest and speakers to the audience.
A very good morning to each one of you, my dear friends, Professor R.P. Sharma, Professor Mahapatra, Professor Lakhotia, Professor Sasidharan, Professor A.K. Singh, Professor Gopala, very distinguished colleagues. I see here, my dear students, ladies and gentlemen, I am extremely grateful to the director of the Institute, Professor A.K. Singh, for giving me the opportunity, this very important occasion of celebrating the 50 years of the discovery of wingless mutant in Drosophila melanogaster. As a matter of fact, the queen of genetics. If you all remember, which you have tried to continue honoring that queen by maintaining a very good system of continuing what Professor R.P. Sharma gave to this institution. And very often some people ask, what Drosophila is doing here at IERI, and how come you give so importance? Being the chairman of the Research Advisory Committee presently for the IERI, I always emphasize that as a matter of fact, Drosophila must occupy the key kind position in our thinking process because it is the queen of genetics. And you all know that queen of genetics, how much knowledge has been generated by working on that queen, and that queen helped in getting so much contributed to the understanding of genetics, chromosome systems, you name it. Today the best of the molecular biology, everything, and application of those systems to the development of the humankind, even management of cancer and so on and so forth in humankind. You can imagine the connectivity with that little fly. And that little fly is the point of celebration which was cared and nurtured by none lesser than, and really experimented upon by none lesser than Professor R.P. Sharma. As a matter of fact, I had the privilege of being colleague when I was director here, Professor Sharma. We worked together, and I always used to admire his devotion to the kind of things that he was doing and the profound impact that it's going to have. Incidentally, I am also privileged that I have known one of the two outstanding speakers here, Lakhotia Sahab, when I was at Banaras Hindu University as the Dean of the Faculty of Agriculture. He was the outstanding scientist working in the zoology department, and I had the privilege of going to his lab and seeing the outstanding work that he was doing on Drosophila. So I am so pleased today that it happens to be a coincidence of this wonderful combination that I am sitting between the two persons who were really so much connected, and I consider it only a major coincidence of the best coincidence to have this opportunity. I will briefly try to see that I can bring out the main contributions and the knowledge that has come through the outstanding role that has been played by my three friends and colleagues whom I have the privilege to introduce. Professor R.P. Sharma was born on February 20th, 1940. Another thing common, I was also born in 1940. He was born in Roorkee, Uttaranchal Pradesh in India. He is a distinguished scientist with an MSc degree in botany in 62, a PhD in cytogenetics. His illustrious career includes postdoctoral research at the University of Stockholm, Sweden, and a UNDP fellowship of Stanford University and Ohio State University in the late 80s. Professor Sharma's extensive employment history includes roles of the Indian Agriculture Research Institute, New Delhi, where he served in various capacities such as geneticist, professor of molecular biology and biotechnology, and later on as a project director at the National Research Center on Plant Biotechnology, the very first center of its kind to be established in this country on biotechnology was here. He also contributed significantly as Chief Technical Advisor, consultant at the Agriculture Genetics Research Institute in Hanoi, Vietnam. And incidentally, I happened to be associated with that. That time I was working for FAO, and the project that he's worked in Vietnam was a FAO project helping Vietnam. So Professor Sharma we have always, whether I am here or I am in Bangkok or in FAO, your outstanding contribution has been recognized, and we have been able to take advantage of your knowledge in the international career also. His research interests, of course, we all know, the mutation research, plant genetic engineering, molecular breeding, and of course biosafety regulations go together. Notable contributions include demonstrating synergistic interactions between gamma rays and EMS-induced mutational lesions, discovering unique mutants in Drosophila melanogaster, and developing insect-resistant transgenic brinjal lines, which of course we in India were unfortunate not able to release and take care of it the way it should have happened. Anyway, somewhere, somebody in the world is making good use of it. So your contribution is always there to help humanity. Humanity is global. Dr. Sharma also played a pivotal role in developing biosafety guidelines for genetically modified crops, and he had received awards and honors for his outstanding contribution, including the Om Prakash Paseen Award in 1999, Science Congress Millennium Plaque of Honor 2003, and the INSA Silver Jubilee Medal in 2013. He is a fellow of prestigious institutions, including the Indian National Science Academy, Indian Academy of Sciences, National Academy of Sciences of India, and the National Academy of Agricultural Sciences India. Dr. Sharma's research areas encompass plant biotechnology, genetics, and cytogenetics, currently serving as an Indian National Science Academy Emeritus Scientist. He was actively involved in coordinating projects for functional genomics of rice. So with these words, Professor Sharma, I want to seek your advice, Director Sir. Would he be making the speech first and then I introduce the speakers as the chance comes? Or should I introduce everybody and then keep sitting and they will come? So what do you advise me? I can introduce everyone and then you expect what is coming to you if you know that what persons we have today. So let me say I am 3 times more privileged today that I have to introduce 3 most outstanding persons. So I have done one already, the other one. Let me mention my other friend whom I knew right from when he was very young and still so active. Even he is as active the way I saw him in early 70s and I see him today, it is the same. The kind of energy Professor Subhash Chandra Lakhotia possesses. And he was born in 1945, is an eminent Indian cytogeneticist and academician whose illustrious career has significantly shaped the understanding of chromosome organization, replication, and gene expression in Drosophila, serving as a distinguished professor of zoology and a Science and Engineering Research Board Distinguished Fellow of Banaras Hindu University. He has been recognized for his groundbreaking work on the 93D gene-long non-coding RNA locus of Drosophila. Dr. Lakhotia, born in Churu, Rajasthan, earned his doctoral degree from Calcutta University in 1970 and embarked on a distinguished academic journey, including postdoctoral studies at prestigious institutions like the University of Edinburgh and the Massachusetts Institutes of Technology. Throughout his career, he has received accolades, including the Shanti Sarup Bhatnagar Prize for Science and Technology in 1989. Notably, he has played a pivotal role in unraveling the functions of long non-coding RNAs since the 1980s, particularly in the context of neurodegeneration and apoptosis ptosis in Drosophila, models of human diseases. Dr. Lakotia's extensive contributions also extend to using Drosophila as a model to explore the mechanisms of action of Ayurvedic formulations, indicating potential applications in suppressing neurodegenerative disorders. With over 200 publications and research papers of very high caliber, and a mentor to more than 35 scholars, his impact on the field are profound, marking him as a distinguished figure in Indian science, which he continues with the same enthusiasm in academia. Is a great honor. Sir, I am privileged to introduce to you to this great audience. Finally, I have the honor to introduce Professor Elias Sasidhara, who is the Director of the National Center for Biological Sciences and Professor of Biology at IISER in Pune. He is a distinguished scientist in genetics, molecular biology, and evolutionary biology. He is renowned for his pioneering work using Drosophila as a model system. He has identified crucial mechanisms governing organ development and growth control during embryonic development. Obviously, you can see whether it is a development in Drosophila or it is in human being, developments through the process of differentiation and gene actions is the pathway which is so common. Sasiji is a founding academic member of the IISER Pune and Ashoka University, has significantly contributed to their growth. He played a key role in establishing the Indian Cancer Genome Atlas and the Pune Knowledge Cluster addressing cancer research and multidisciplinary collaborative initiatives. Now you see, from Drosophila to cancer. Recipient of the CSIR Technology and SS Bhatnagar Prizes as Professor Lakotia, J.C. Bose National Research Fellowship and Associate Member of the IMBO. Sasiji has the international recognition and he speaks to his excellence and the details that we know about him in life sciences research is something unmatched. Notably, he is the first Indian president of the International Union of Biological Sciences. Let us give a big hand. This is the first Indian to be the president of the International Union of Biological Sciences. During the COVID-19 pandemic, Sasiji actively engaged in various fronts to mitigate its impact, from epidemiological data analysis to indigenization of the reagents for RT-PCR kits. And you know, COVID is, you never know. It is already again being talked about in the evening news. When you hear it again. Beyond research, Sasiji has held leadership roles in apex committees focusing on education, research, policy, and outreach. He has trained over 10,000 teachers, mind you, 10,000 teachers, and each of those teachers even train 1,000. You see, the multiplier effect from Sasiji is amazing, amazing. He organized career workshops for scientists and is steering an international project on climate change education. And today, if anything is important, most important, climate change is most important. And again, that is his major concern. Professor Elias Sasidhara emerges as a distinguished figure, significantly contributing to biological sciences, science education, and collaborative research in India and globally. Ladies and gentlemen, I am so honored and I am so privileged to introduce, and I myself get invigorated really to have introduced each one of you. Thank you, my dear friends. You know what is coming to you. you will be getting the best of science today.
Thank you, sir. We are now eager to hear from our chief guest, Professor R.P. Sharma, and gain a deeper understanding of the journey that has led us to this momentous occasion. So, ladies and gentlemen, please put your hands together and invite Professor R.P. Sharma to share with us his reminiscences of the discovery.
Good morning, Professor R.B. Singh, Professor A.K. Singh, Professor Lakhotia, Professor Sridhar, Gopal, all my old colleagues, students, and friends. I feel myself so privileged and lucky to be standing here. If I recollect, the first time I stood here was in 1965, when Dr. Swaminathan had organized a Mendel Symposium, 100 years of Mendel's discovery. It has been a long journey, and I cherish it. Dr. Mahapatra has, in great detail, talked about my work. But I think the individuals are important, but their associates are much more important. I'm lucky to have Dr. Mahapatra as a student, as a guide, and at times as a teacher as well. His understanding of the subject, I know most of us have learned while doing. I know the experiments getting failed, and in the night, Dr. Mahapatra was feeling rejected, and then we are supporting. Because we didn't have any expertise in this institute in molecular breeding. And we started from the scratch, starting from the DNA isolation. Now it looks so easy, but those days, in early 90s, it was a difficult task. And Mahapatra is an example how you can fight with the odds and then succeed. And I'm really thankful for him, for his contribution. His contribution to the NARS as a whole, his contribution to IRI, and his contribution basically to the subject of genetics and plant breeding and biotechnology. When I was asked, in fact, I was discussing with Dr. Mahapatra many times about this mutant, and then he read himself, and he found it pervading almost everywhere. The total biological system is pervaded by this particular mutant and this particular gene. And then it is his idea that we should celebrate, because it's going to be 2023 is going to be 50 years of his discovery, and therefore we must celebrate the Golden Jubilee. He proposed it to Dr. A.K. Singh, and both of them agreed, and that's how you have this function today organized here. I'm a brief presentation of 10-15 minutes. We'd like to give you the basics of how we have moved from and how I perceive this particular mutant, and the rest of the scientific part you are going to listen to from our learned speakers, Dr. Lakhotia and Dr. Sridhar. May I have the first slide, please? Can you tell me how it is done? This forward, reverse, and forward. This one. Yeah. I dedicate this lecture to Dr. Swaminathan. That's how he looked in 60s. And I thought that when I'm talking about 60s of work, I must have an appropriate photograph of Dr. Swaminathan. But unfortunately, he is no more. we had the condolence in this meeting and all over the country and around the world. His total contribution, I've seen him because fortunately I got the opportunity of doing my PhD with him, and therefore I've looked from very close quarters. The excellent teacher. In fact, some of you I brought to, there will be some, in his class used to be overflowing. And generally he used to take classes because he will visit during meeting at certain day time, so he will take classes in the late evenings or early mornings, and you will find all the time his lecture room will be overflowing by students. Impressive his lectures used to be. Excellent teacher, excellent researcher, which all of you know, the way he has transformed the total agriculture in this country. Above all, a great human being. I've never seen him ever losing temper. He will listen to you, he will help you, and that's the greatness of this great person. And therefore, this particular 10-15 minutes of presentation are in his memory. When I joined this institute in 1962, that was the time when all of us were concerned about the atomic energy. Destruction which was caused by the atom bomb in Nagasaki and Hiroshima. And therefore I took from the literature, Atoms for Peace was the address given by Eisenhower in the United Nations on 8th December 1953. In 1957, the International Atomic Energy was established. Swaminathan returned in 1954, joined this institute in 1954, and was completely conversant with what is happening around in the world in terms of peaceful usage of atomic energy. And he replicated those in India in terms of starting direct and indirect effect of radiation. For that, he established a Gamma garden. Many young people would not know, but you must read, it used to be the NAC complex where it is, a Gamma garden with thick, about 1 meter round, 100 meter radius, 1 meter thick walls. The radiation source used to be at 6 feet underground. When you want to take the radiation, you push it off, the radiation source will come and will radiate your material, and then you put it back in the ground. So that's that Gamma garden. Fortunately, afterwards, techniques developed and Gamma garden was converted to Gamma cell, and most of work later on subsequently was done on Gamma cell, where the thick lead covering will protect the environment from outside. And that's where, when the Drosophila lab was developed. It's a story, very small one. When he wanted to study the indirect effect of radiation, he asked his student Satya Nirula to go to Hyderabad and pick up few techniques of butterfly, etc. from Dr. O.S. Reddy, who had just returned from US. And that's how the Drosophila lab was started. I would like to have some, it takes a time. This is the work which we did on the indirect effect of radiation. It was published in Science. So I got a moral and psychological lift. Immediately the first year of my career of joining, we ended up publishing in Science, this paper. As you see here, the induced mutations by radiating the food and then feeding it to the flies and then getting the cyclic lethal produced, there is an increase in the lethal mutations. But it is not very significant. It is, but then we concluded that in 3 different experiments indicated that irradiated media have a mutagenic effect. 6-reactive lethals of the order recorded in these experiments may occur in the control population, it is clear that the effects produced by the irradiation of the medium are small and can be demonstrated only in relation to control of comparable strength. So afterwards, a lot of studies have been done in this area, and now this the whole thing, this is where it stands. If you go and read our Atomic Energy Commission report, you will find, and this is I have taken from FDA, has approved a variety of food for radiation in the United States, including beef, pork, and poultry, shell egg, and shellfish, crustaceans, fresh fruits and vegetables, leafy vegetables, seeds for sprouting, spices, and seasoning. These are the ones which are now being routinely being irradiated by a particular dose so that these can be commercially exploited. From here, we had been doing mostly that type of radiation, but then time came when we started. Dr. Swaminathan asked us, do lesions produced by radiation and chemicals interact? And that's where we studied those in Drosophila, we studied in Barley, we studied in Aspergillus, and very interesting story, and that shows how encouraging Dr. Swaminathan had been also. I say, sir, I do not know Aspergillus. He says, go to Obed Siddiqui in TIFR Bombay, he is handling Aspergillus, and that's how I spent 2 months in TIFR, learned those techniques, and came back and started working. So these are the 3 systems on which I was working simultaneously: Drosophila, Barley, and Aspergillus. And we concluded that when you combine physical and chemical regions, you get a synergistic interaction. But soon, EMS was turning out to be very effective, and we start producing mutations. And this has been my life's mantra. If you want to understand a gene and its function, mutate it. And that's what I followed all through. And I'm going to give you a glimpse of that, what all we have achieved. The first thing, the behavioral mutants which we isolated were shakers, hyperkinatic mutants, paralytic mutants, line-dependent homosexual mutant. I got this stimulation from this gentleman. All of you, students of genetics and molecular biology, might have read R2 locus of T4 phase. Benzer was deeply involved. He identified, he could define recones, mutones, and cistones. And then afterwards, he asked himself, what is next? Because the system which was handling was not really, it has come to a kind of dead end as far as he was concerned. So he was looking for, he had 2 daughters, both of them behaviorally completely different from each other. Then he started asking, can I study the behavior? You cannot study behavior certainly because they are socially certain human beings. Then he chose Drosophila. And he isolated, this is the brief summary I am giving. He had studied fine structure of gene autolocus, genetic code, behavioral genetics. He founded the area of neurogenetics. So if what you see in the literature, neurogenetics basically arose from Samuel Benzer. Isolated mutants for learning, optomotor, hyperexcitable, dropped dead. And from here I got distribution and then started looking for behavioral mutants. And one of the behavioral mutants if you see, it's a very interesting story here. You have Benzer counter-current distribution method. Benzer was basically a physicist. And when he developed a technique, he gave that name counter-current distribution. I was excited. What is counter-current distribution? And we went into the literature. It was nothing but light phototactic response of Drosophila. He developed a chamber where he will put the lights towards Drosophila will move towards the lights. They are positive tactic. If they are not moving, they are negative phototactic. And he isolated mutants both for photoelectrics as well as for locomotion. I repeated that experiment here and isolated the mutant, which is, I thought, from many. I will just put this for your knowledge. Counter-current distribution protocol helps in identifying a sluggish mutant with a very poor photoelectric response. Now, a detailed analysis of this mutant ultimately told that this is associated with X and third chromosome translocation, slows light-induced homosexual activity, yellow light induced maximum sexual activity, and the moment you change the light spectrum to 600 nanometer, it instantaneously terminated the sexual activity. So that was very interesting. One day in the morning I came in the tube, I saw a lot of flights, lot of activities there. I saw from close quarters, and I find that this activity was not innocuous. It was the males were sexually aroused, and they were moving each other. And then that's what it was. These are all males. They are performing the sexual activity both of attracting and also pseudo-copulation. Most of the sexual activity in the insect is pheromone drive. It is the light-induced drive and instantaneous. The moment you put 450 nanometer light, the males will start following males. The moment you put it at 600, they will immediately separate out as though nothing has happened. This is, unfortunately, this mutant is lost. Otherwise, I sent it to Dr. Obed Siddiqui at the time, the paper which we published in Experiencia. Dr. Obed Siddiqui was I consulted him on it, and I thought that his lab has not yet reached to that stage that one could do start that recording of the single cell from the retina. But this is one of the examples which I thought you would like to know how Drosophila can be useful in various kinds of let's come to for today's exposure, developmental mutants. Upon spiny legs and wingless, some of this we had mutated, and the wingless is the one which is the topic for today. So I will like to this is a story which is interesting in the sense that we were inducing sex-linked recessive mutations. We were studying them. We were studying the temperature-sensitive mutations as far as sex-linked concerned, and got quite a few developmental mutants. In one tube, we found that along with the lethals, there are some flies which are wingless. We thought that we have, if you know, in Drosophila, there is you can get a lot of abnormalities in the wings, but then when you started doing genetics of it, we find that excuse me, we find that from the same flies you get some which do not have any wing. There are some flies which have one wing. There are some flies which have both the wings. Now, when we started studying them, we find that they are genetically all these flies are same, because we separated wingless, crossed them separately. We separated one wing, crossed them separately. We separated two wings from that cross and crossed them and found them that from each of these three crosses you get all the three phenotypes again. So genetics then ultimately was simple, that genetically they are same, similarly governed. It's only the penetrance level which is different. There were a lot of discussion at the time. We said the discussion that involved was Peter Bryant, Irwin, Peter Lawrence from the Cambridge. Then we had Garcia Bellido, and we have Borata. These were the ones that they were in Obed Siddiqui's lab. We used to move And quite a few, these people, they were saying that is a homeotic mutant. We were saying it is cell death followed by mirror image duplication. And now we know that it produces cell death in the imaginal disc, which leads to mirror image duplications. And of course now we know it's a segment polarity mutant. This is what the mutant which was initially isolated. There are no wings, but they are halters. But you can have flies without wing, without halters. You can also have flies with no wing, but both the halters. Halters are balancers. So if you also we got some two-wing flies with halters, no halters. And it was an interesting experiment. Halters are responsible for balancing in flight. So when we took these flies without halters and put them in the flight, there was gliding. They were not flying. They glided out and then fell. So many people earlier do the experiments by scissoring it out. Remove the halters and then see what do they do. They say only gliding. Here it was a natural process. There are no halters and the flies will not fly. This is with the rare frequency. You used to get whole this. This is notum, thorax. All notum is completely missing in this. Only so you have only head, ventral part of it you have to attach to the legs, and you have abdomen. So this is the first report which when I got this. This is the first report which we sent to Drosophila information. Acha, okay. This is the first report on the wingless, which we reported in DI Drosophila Information Service in 1973, and that's the what we say very golden jubilee. It's a 50 years of the discovery of this mutant. This when we studied it in greater detail. This was the detailed studies were later on published by in developmental biology in 1976 with Professor Chopra mentioned in this. It was based on. You see, this is what the flies they look like. They don't have wing. The scutellum is completely deformed, and here most of the bristles etc. are missing. Here one wing, one of these notum is developed, otherwise completely damaged, half of it or even less than half. This is his half is upset. In Drosophila, in fact, you have imaginal disc, and during metamorphosis they develop into full organ. This is the wing disc. This has the halter, this is the notum, and this is the wing. The pouch which will come out and that will form in the wing. These are the leg disc and these are the halter disc. This is you can take out of the larvae and compare and study what they are. This is what it will look. If you have a wingless fly, this is how your wing disc. This is the normal wing disc. This is the one where this particular part of the wing part is completely missing. So this we studied. Mirror image duplication we studied. Mirror image duplication we studied here in this, and in this these are the ones which are halter and very earthly study. What we have done, we collected a large number of flies and then we studied the thorax, and you will see here. Each of the fly here, they are having, these are the mirror image symmetry from where we have studied in the visual discs. So you have all developmental defects in the imaginal disc and ultimately converted into metathorax or thoracic disc. Next. Therefore, where do we stand now? This is a gene symbol which is wingless, a protein coding gene, and sequence location is on second chromosome long arm at this sequence. Cytological map is at this one, the band in the salivary gland, and recombination data is chromosome 2, 30 map unit. So this is the genetics part of it. Then two things happened which made this mutant meaningful to rest of the world. First thing is, this is a paper produced by Nelson Vollard and Eric Wichhaus, and they studied the lethal allele of wingless, and they found that the segment polarity is depicted by this is the normal larva, the ventral part of these are the denticles here, which are typical. Those of you who are not interested in it can only know that this is goose and hedgehog, these are different mutants, and you find that this is the anterior part, this posterior part, it gets deleted and then gets its mirror image duplicated, and that's what has happened in this particular mutant. Next please. These are the three persons, Christian Vollard and Wichhaus, from this work, and E.B. Lewis, I think all of you know, the student of genetics would know, these three got Nobel Prize in 1995 on this. And this particular mutant therefore was, they characterized it as segment polarity mutant. All of us, starting from nematode to human being, all of us, our bodies are segmented. In the embryonic stage itself, the segment is decided. If your segment polarity is not properly decided, it leads to malfunctioning and aberrations. So this particular gene now, as we know, affects the cell pattern, its segment polarity, it produces morphogen which can be signal transfer from a cell, within a cell, from a cell to another cell, and ultimately what Dr. Sridhara will be discussed will lead to aberrations and cancer. The another important observation which was done by Regiswick was that when Drosophila human Drosophila genome sequence was known, mouse genome sequence was known. In mouse there is a gene INT that is responsible for mammary tumor. Where the gene is when the mouse, when the virus gets integrated in the genome, it's decided by the INT1. Incidentally, INT1 was shown to be completely homologous to wingless, and that gave a flip, because this was the first mutant where a phenotype, a gene which has a phenotype is causing a cancer, and that's where then things proceeded, and I'm going to. That's where the change from wingless, a new name came, that is WINT. And those of you are interested in following biology in detail, beside plants, put on your Google WINT signaling pathway. Just put WINT and put any organ of human body, whether it could be heart or it could be brain or it could be liver or it could be kidney or something, and you will get the latest publication. Because this particular gene is having effect in almost all the human organs on their development. I am a heart patient, and therefore I was curious to know what it is doing in cardiac area, and therefore I am going to give you some examples of that. So what has happened is since it was wingless and now it was going to humans and rats, etc., it was thought that, and it had homology with INT, so the new name was generated, which was called as WINT, what W from wingless and NT from INT. So now we know, we call it as wingless also we call it, but we also call it WINT. There are, in human beings, in Drosophila there are now 7 copies of this gene. In nematode there is 1 copy of this gene. In human being and mouse there are 19 copies of this gene. This scattered in the whole genome. not a clustered at one place. It's scattered in the whole genome and doing all kinds of functions. 19 copies of Wnt gene in. And what this gene basically does, it decides the cell fate. The moment in the pattern, a cell is placed in a particular area, its fate is decided. What organ is going to develop further. Whether it could be anterior, posterior, dorsal, ventral, anything. It's the fate decision this gene helps. Cell proliferation, it helps, it decides. Cell migration, cell polarity, cell death. In human, 19 Wnt proteins have been identified in adults. Wnt function in homeostasis and inappropriate activation of Wnt pathway is implicated in variety of cancers. This is the pathway through which this particular gene works. It has the canonical pathway, it has non-canonical pathway. Planar cell polarity, calcium dependent pathway and Wnt canonical pathway. And then it goes, it is recognized by the receptor of the cell membrane, then it goes into the cell and then it goes into the nucleus and then it activates the genes which are basically responsible for various kinds of functions. I have taken only one example and that is Wnt6. This you can have for all the genes. In Wnt6, this particular gene, the Wnt6 is involved in cardiac injury, kidney fibrosis, lung tuberculosis, Rettel syndrome, glycostone, colorectal cancer, gastric cancer. Details one can retrieve from the literature. Beta-catenin, which is a cholinergic pathway, it is several Wnt beta-catenin pathways. Inhibitors are now being discovered and they are responsible for cardiac diseases. The one most important thing which you will see, the human heart, you will have the enteroventricular nodes and you have the sagittal nodes. These are the nodes which are decided, the cells are decided much early in embryogenesis when heart is formed. And then these are the cells which have the pulsating movement, rhythm, the electric current, impulse, action potential. And that's the one which migrate into ventricles and then the heart contracts and throws the blood. Now these cells here are the ones which now have been shown to be regenerated. You can regenerate them using by manipulating the Wnt pathway. This would mean that if you have a heart disease which the pumping or the electric current generation has weakened or is aberrant, then you can have these cells replaced or regenerated. That's the kind of science which is going to develop and coming from it. The de novo pacemaker cells, natural pacemakers which we have are being replaced by artificial pacemakers by the doctors. But if you can have the de novo pacemaker cells exhibit hallmark features of native cardiac pacemaker cells attesting to their therapeutic potential for cardiac rhythm related diseases. So once you have that kind of thing, then you will have the cells which will be proliferating. You can regenerate, you can replace, you can make them function for more time. So in my opinion, that the gene which we have isolated is so innocuous looking at that time are now turning out to be so crucial in human diseases of almost all the organs. I put here only cardiac. Dr. Sidra will put over the cancer and all that, and you will see. So therefore, to conclude, we have Drosophila wingless, which we isolated, has gone to its midst. 50 years are past, but is still counting. Many more things are going to come. In fact, your this particular gene is going to provide you the therapeutic means of various kinds of ailments in human beings. The Wnt signaling pathway is an evolutionary, Dr. Sridhara is going to discuss. I think I will stop it here, and thank you very much for your patience listening. Thanks a lot.
Indeed,
Thank you, sir. Thank you, sir.
indeed a very wonderful deliberation. So your enormous dedication and determination towards science is a true inspiration to most of us. So we feel privileged to have heard directly from a person who has played a very vital role in this significant discovery. Thank you so very much, sir. It's time to hear from our keynote speakers. May I now invite Dr. S.C. Lakotia, Distinguished Professor, Department of Geology, Banaras Hindu University, Varanasi, to deliver his lecture on a very interesting and relevant topic, Historical Perspective of Fly Research in India.
Okay, good morning everyone. Professor R.P. Singh, Professor A.K. Singh, Professor R.P. Sharma, Professor Sridhara and Professor Gopal Krishnan and all the distinguished Professor Mahapatra online. It's a great privilege and pleasure for me to be here to celebrate the golden jubilee of Wingless discovery in Drosophila. It being an historical movement, I thought I'll briefly recapitulate history of Drosophila research in India, especially for the benefit of the young audience, to see how Drosophila has contributed and how it is expanding its wings all over the country and in fact across the discipline world. Professor R.P. Singh said it's queen of genetics. The Drosophilists call it at Cinderella of genetics. That's the small dear Cinderella. And this work, this insect was introduced to genetics by Thomas and Morgan, an embryologist, and who started in 1905 his lab, and then slowly over the years in 1910 he got the first white-eye mutant, and then it went on. If I can move to the next slide, okay. Yeah, the history in Indian science where Drosophila introduced in India, I'll just briefly summarize here that the first group was in Calcutta. Professor S.P. Chaudhary, who joined after doing his PhD with H.J. Muller, joined Calcutta University, and then with help of J.B.S. Haldane, I'll narrate that story a little later, started Drosophila genetics. Then he moved to Banaras in 1960 and set up a Drosophila lab there. Besides mammalian chromosome, mammalian cytogenetics group, he continued with Drosophila, grasshopper and so on. And then Dr. A.S. Mukherjee, my supervisor, joined Calcutta University in 1965 after his PhD with Kurt Stern and the postdoc with Behrman. He started the genetics research unit in Calcutta, and that's where fly work continued in the 60s in Calcutta. And then the Varanasi group, as I said, Professor Chaudhary started in 1960. Then Punjab group again in 1960 by Professor G.P. Sharma, and then of course he has his colleagues and students, Dr. Ram Prasad, Dr. Narda, K.S. Gill, Miglani, whose school has continued later on in Haryana, and then the Mysore group, Professor Rajshekar Shetty, who was trained with Dobzhansky, started in 1960 in Mysore, and his group has continued. I'll briefly trace these groups, how the lineages have gone on till current times. And then the IRI group started in 1960, which partly was directed here. Professor V.L. Chopra was one of the first trained Drosophila person, and then of course we had Professor R.P. Sharma continuing in that work. Then Hyderabad group, Professor O.S. Reddy, who had, was also trained with Orbach and joined Osmania University in 1963, started Drosophila work there with M. Sanjay Rao, his student, continuing for some time. And then the TFR group, Obaid Siddiqui, started using Drosophila in 1970s, although he joined TFR in 1960s, but it was only after he went to the Samuel Benjer's lab that he got to use Drosophila. I'll try to trace these stories slowly. But before that, let's get into this pedigree. As in genetics, we often talk of pedigree, and we all can trace, all the pioneer geneticists in India can trace a pedigree to, I'm sorry. Okay, I think this is the point. Yeah, to Professor Thomas and Morgan, his student Sturtevant, Muller, and Stern was his postdoc. Dobzhansky was his postdoc, and so these are the three main lineages from which Indian Drosophila workers started. As I said, Professor Raichaudhuri was Muller's student, did his PhD with him, and then his student, he taught A.S. Mukherjee as his master's student. And his PhD student J.P. Gupta at BHU, who started the Drosophila work. I happened to be also kind of not a direct student of Professor Raichaudhuri, but he was my examiner both at a master's and at my PhD. And I did my PhD with Professor S. Mukherjee, so we can trace our lineage. Then the other group was the K.S. Gill's group, who is in Punjab, his Ravi Prakash, and then that's in continuing. Obaid Siddiqui with Benjer, Delbrook was Sturtevant's postdoc, and of course Muller's student Pontecorvo, from which Obaid Siddiqui learned Aspergillus work and from Benjer Drosophila work, and then the lineage continued. The other side, Orbach, again Muller's collaborator and student, V.L. Chopra and O.S. Reddy. I'm sorry I don't have a photograph of O.S. Reddy. I don't know if somebody has it. I will be happy to have an ad in this blank space here. And then the other group that started my student, which is continuing till date, was Professor Rashekar Shetty, who set up it, who was Dobzhansky's and Dunn's student. And then Professor N.B. Krishnamurthy was Rashekar Shetty and had also training with W.S. Stone, who was a postdoc or a PhD student of H.J. Muller, and then Professor Ranganath, who is continuing. And so this is how the initial groups in 1970s were in Drosophila. Now, one by one, let's say J.B.S. Halden, how did he contribute to Drosophila research in India? This is something which is not very widely known, but Professor Raichaudhuri, after he came back from Edinburgh after Second World War, joined Calcutta University, could not continue Drosophila because you can't have air-conditioned lab and other facilities, so he started working with grasshoppers because that is something that he had learned from another great cytologist scholar, and he also started working with some cancer cell cytology and so on. J.B.S. Halden, when he was moving to Calcutta in 1950s, and he happened to be a PhD examiner of Raichaudhuri, he knew that Raichaudhuri is in Calcutta, and he met him and found out that he is not working with Drosophila. Now this is something very interesting. J.B.S. Halden then talked with Nehru, the then Prime Minister of India, that he would like a Drosophila lab to be set up in Calcutta University, and Raichaudhuri can be consulted as to what will be needed. Then the Prime Minister talked to the Vice Chancellor of Calcutta University and Chief Minister of West Bengal, B.C. Ray, to find out what can be done. And Calcutta University then, in consultation with Raichaudhuri, set up the Drosophila lab in the old Science College building of Calcutta, where I was an MSc student later on. And then Professor Raichaudhuri was waiting for Halden to come and take start working with Drosophila, and then Halden said, "This is for you." And that's how the Fly Lab started in India, and that's the beginning. And this is a remarkable story which tells that how things can happen if there is a proper interest and proper drive given to this. And that's what happened, and actually this was J.B.S. Halden, of course, and this is the Calcutta University, the then head Professor J.L. Bhaduri, Professor Girish Chakravarti, then M.M. Chakravarti, and Professor Raichaudhuri, and Halden's wife. I happened to be taught by all these three people during my masters when I was a student in Calcutta. And actually, I worked in the lab for our MSc in the lab where Professor Halden had set it up, and then moved to the new building and many things that I'm lucky that during my PhD I had the privilege of working in that lab. And Professor Rachudri, of course, left a long legacy. His mentorship was later on taken by Dr. A.S. Mukherjee when he joined Caltech University in 65. Having done his PhD with Kurt Stern, and in 57 he had his master's project on Drosophila ananasi, which was something that Professor Rachudri introduced to India other than melanogaster, and that has continued. And this is Professor Rachudri, his student in Banaras, where he set up a Drosophila lab, and there was, most of them worked with flies. Professor Gupta here, Professor B.N. Singh, who was later on, and Mercy Jacob, she worked on radiation genetics in Drosophila and then moved to DNA repair in mammalian cells, has been my colleague, and Rajiv Raman, who was, so this was a vibrant group he set up. And of course, me, well, later on when I joined, my colleagues, Professor J.P. Gupta and Professor B.N. Singh, who both of them had their PhD with Professor Rachudri on Drosophila. And then, of course, that's the legacy at BHU is Professor J.P. Gupta and Professor Rachudri B.N. Singh. B.N. Singh continues as a distinguished professor in BHU doing population genetics and Drosophila ananasi. And his student, A.K. Singh, also continues to be at Drosophila. So, and then of course their students have set up several fly labs in the country at various places. And this is of course, in genetics we talk of F1 and F2 and F3. So I can claim myself to be an F3 of Professor Rachudri, and this was a meeting in Udaipur where Professor Rachudri, his student Dr. Mukherjee, and my students, two of my first students are here. Then Dr. Mukherjee's student, Professor Rachudri, with Kitish Majumdar, who unfortunately passed away a few years ago, was also there. So, you know, this is interesting that in fly community, the community remains very well knit even after generations and so on. And this, of course, myself in 1968 in Calcutta, two of my colleagues who are now in US, and Professor R.M. Chatterjee took over after Dr. Mukherjee passed away very prematurely in 2000. But then after he retired, Caltech Fly Lab is almost closed now, unfortunately. That's what has happened. The other group was TFR group, which I said Obed Siddiqui set up. Obed Siddiqui was trained in Aspergillus with Pontecorvo, who was Muller's associate. And of course, he learned fly with Samir Benja that Professor Sharma had mentioned, that is the father of behavioral genetics in Drosophila, moved from the R2 locus of T4 bacteriophage to fly, and Samir Benja has contributed enormously, and that's what the dual lineage that Professor Siddiqui had. He set up the remarkable molecular biology unit at TFR, which now has NCBS, that's where Shashidhar is now currently director of that institute. And of course, they also have a TFR Hyderabad group where they're continuing with fly work with younger colleagues. And today, the fly has spread all across India, from Nagaland to Gujarat, from Kashmir to deep south. Everywhere there are labs with lots of young people who have joined in the last 2 decades, various ICERS institutes, central universities and regular universities, that the fly community has. Now there are about, I should say, 140 to 150 fly researchers across the country. And this is something very remarkable. And one can see a glimpse of this. The recently held Indian Drosophila Research Conference that has started in 2013, now it has this 2023 edition. This is only less than half of the fly workers were there attending, but it's a huge group. And this group continues to grow, and that's where the importance of fly is. And this is now briefly about Drosophila at IARI. Why should IARI Agriculture Institute should have Drosophila work? And this is something remarkable that the far side of Professor Swaminathan, that when he was interested, as Professor Sharma had briefly mentioned, about the radiation-induced mutations and radiation-induced agriculture products, whether they could be mutagenic. And Drosophila was found to be one of the best model. And so that's how it started. And as Professor Sharma said, that Dr. Nirula was sent to Osmania to learn basics of rearing of Drosophila. Professor Chopra later on joined, and that's how Professor Sharma set up the fly lab here, an active fly group. And Shanti Chandrasekharan, his student, unfortunately we lost her very prematurely, who had done remarkable work on fly, and her student Prachi is continuing here, and I hope this grows up at this place also, because see, basic research and applied research, we make sometimes, I believe, artificial distinctions. Any research has an application later on at some time or the other to come. And Drosophila continues to be a remarkable system to find out a variety of things which may and which would have great relevance to agriculture, to livestock, and whatever it is. And of sorry. This is the great paper that we are celebrating, the wingless mutation that was discovered as part of EMS mutagenesis, and which happened to be there. And then, as Professor Sharma mentioned, that they published a more detailed work in 1976 in Developmental Biology, a very leading journal. And then, of course, as he had shown that it had a varied phenotype, and it became a wonderful model for subsequent studies in a variety of things, which I am sure Shashi will talk about, how the wind signaling has become so important in basic as well as applied aspects. And of course, they had authored a very good review in Current Science, which talks about what this group has contributed, what kind of mutants it had discovered. So this is a very good summary, and anybody who is interested in looking at the historical perspective of a fly group at IERI, this paper provides a very good summary of that. And then, of course, briefly about myself, what I've been doing. At BHU since 76, and as I said, my lineage, dual lineage, I continued, and I had initially set up a fly lab in Bardhan University when I was there 71 to 72, then at Gujarat University 72 to 76, and then from 1976 I'm at BHU. And some of my students who had gathered for some chromosome, some functions we had together, and my major research areas are gene expression, chromosome replication, stress biology, heterochromatin, long non-coding RNA biology, tumor biology, and Ayurvedic biology. So major contribution that I could have been made, which I think have had important thing, was this doctoral work, where we showed that the Haber-Malex model of dosage company was established, confirmed, which Dr. Mukherjee had proposed earlier with Biermann's lab, and then that this was a cellular autonomy, that even if there is a single male cell in a gynandric Drosophila, its sex chromosome will be hyperactive. That was an important discovery that we made. And then, of course, I showed that heterochromatin doesn't replicate, beta heterochromatin transcribes. This was again something very unbelievable in 1973, that heterochromatin is transcriptionally active, but that's what we showed, and now we know it's very important. Then replication of different replication units in chromosomes, how active replicons are organized using DNA fiber rotography. We measure the rate of fork migration, the size of active replicons of different kinds in different cells. And then the HSR omega, the 93D gene, the developmentally active stress-induced non-coding gene, to call it non-coding in 1982, and then to follow it up at that time was really difficult, because it would be typical selfish or junk DNA. But I never believed that there is anything like junk DNA or selfish DNA, and followed it up. And today we know that the non-coding RNA is everywhere. Everything we talk about, whether it be human disease, whether it be development, whether it be agriculture product, or anything, it's non-coding RNA which is in the forefront. They are the major regulatory circles at every level. And then we discovered the HSP60 proteins in Drosophila and the genes that are associated with them. We established Drosophila as a model for Ayurvedic biology studies, and that Amlaki Rasa and Rasindu can be potent suppressors of neurodegeneration, and this is something that I think Ayurvedic community should follow it up, and we should contribute the herbal products. Instead of using pure herbal products, it should be the typical Ayurvedic formulation, the rasayanas, which should be tried and followed up. And of course, recently we got into tumor biology and we could show that heat shock proteins, the constituted heat shock proteins are important for establishing a cell on the tumorous pathway, but HSV70, the stress-inducible HSV70 seems to be crucial for that cell becoming malignant. That's what we have been able to show. This next slide is not moving. No, there should be one slide in between. What happened? The slide which I thought where we will communicate our greetings to Professor Sharma from the fly community of India on this remarkable day and wish him all the best for continuing to be active and continuing to be guiding the young colleagues. And with that, I thank you all for giving me this opportunity, the organizers here. Thank you.
Thank you, sir, for taking us through the fascinating journey of Drosophila research in India and world over and introducing the legendary personalities of fly community to our young students. So moving forward, we have with us our second keynote speaker, Professor L.S. Seshidhara, Director NCBS Bengaluru, another eminent personality with profound expertise in the field of Drosophila genetics. Sir is going to deliver a lecture on the topic 50 years of wingless from fly wing development to human cancer. So may I invite you to kindly come forward and deliver the lecture.
Thank you very much for this opportunity to be here. It's really an honor and privilege for multiple reasons. One is, of course, we are celebrating 50 years of the discovery of wingless mutation in Drosophila, but basically wingless gene in Drosophila, and the mutation is only a way to discovery of the gene. Second is also, where we are doing this. IRI, Pusa Institute is the one which actually made what India is today. Without food security, we could not have achieved any other, achievement that we are celebrating every year in India, whether it is in space, whether it is in IT sector, whether it is in sports. It's all because we had food security, and food security is because of this particular institution led by Professor M.S. Swaminathan and others, and without which we would not have been standing today with pride and confidence. Today what I'll do is to very quickly take you to the, what some of the discoveries have already been mentioned by Professor R.B. Sharma. So, students may ask, oh, it's one mutation and of a particular gene, why do we need to celebrate? The two reasons. One is, of course, the impact. Impact is always felt few years later. It may not be on the day when it is published. Although sometimes one can actually predict the impact of the discovery on the same day, like for example DNA double helix. When it was announced in 1953 in the Nature, both the discoverers and the readers knew the impact of that discovery. The last sentence of that particular paper, which talks also about semi-conservative method of DNA replication, which is the most important aspect of that discovery. It's not simply, what is the structure. The structure leading to its particular emergent property is the semi-conservative replication of DNA, which is the most important aspect of DNA double helix discovery. So similarly, there is one particular sentence in the Drosophila newsletter which Professor R.P. Sharma has published, and the last sentence I'm going to read out later, which basically also makes it the impact of the discovery at that time itself. In a way, it also talks about the foresight of the discoverer in the process, right? So very often when we celebrate any specific event and retrospectively look at, we are not simply celebrating a particular event, impact of that particular event. It is also the process of science. The process of science requires very good observational skills, very good experimental design skills, and analysis of the results, right? All these things are to be combined to make impactful discovery. And all of us, when we do research, particularly myself, removing all others in the audience as well as outside friends, we do what is called incremental science. And we add few things here and there to what is already known knowledge. But some people actually make some kind of a landmark discovery, and that landmark discovery is behind that is the fantastic, the method of science that we need to learn. So every discovery is the celebration of methods of science, and this is precisely one such discovery, is the discovery of the wingless mutation and its interpretation of the mutation. And Professor Sharma showed the images of very different phenotypes. In our jargon we call it as penetration. You have both the wings are lost, both the halteres are lost, or one wing, one halter, two wings, one halter, two wings, for null combinations, because there are two wings and two halters. And there is penetration, and this actually the paper discussed a lot about the penetration itself, and there are a lot of implications of why there is such a diverse phenotype for a single gene, because he also talks about that there are three different versions of this phenotype, but they all belong to the same locus, and the fantastic complementation genetics they have done. And again, because it's a mutation in the regulatory region of the gene, and now we all know, in those days nothing was known about the structure of eukaryotic gene, and because it's a regulatory region, it can actually have a very different effect on the levels of expression and the time of expression and the spatial distribution of expression. Is not simply about protein is expressed or not expressed, or is functional or not functional, because if it's a mutation in the coding region, right? And that's what leads to this variations in the phenotype, and each one of those variant phenotypes is itself a story about how development may happen early during embryonic development. So I work on Hox genes. I started my work on Hox genes around 1991, and by that time Drosophila embryonic development was well established, and this was the peak season for developmental biology using Drosophila as a model. Every issue of Nature, Cell or Science on those days used to have 3 to 4 papers at least in the area of Drosophila developmental biology. And in the series of hierarchical regulatory events that happens early during development, starting from no information in the single cell embryo to generate information that where is the head, where is the tail going to be, where the head going to be, where the trunk going to be, where the thoracic segment going to be or abdominal segment going to be, and where different appendages along the body axis, particularly anterior posterior axis, which was well established at that time, and subsequently dorsoventral axis was established. Various different concepts in developmental biology established in this Drosophila as a model, and most of this, it's not about gene conservation alone. Developmental mechanisms are conserved. For example, use of morphogen gradient, where it, like Hedgehog gradient, Wnt gradient, TGF beta gradients, they are highly conserved developmental mechanisms. The hierarchy of events, the transcription factor turning on one other next each others, is also well established. And one such family of transcription factors called Hox proteins and coded by Hox genes, and depending on where they are expressed, different parts of the body are established, and they are again highly conserved from Drosophila to human, whereas Drosophila has 8 Hox proteins, humans have 44. Like what R.P. Sharma mentioned, there is one wingless gene in Drosophila, there are 8 such genes in human. It's also a lesson in the evolution where gene duplication and divergence lead to more complexity in the body organization. Sorry. So very quickly, the Drosophila has 2 parts in the life cycle. All of you must have studied in the school textbook. There is what is called hemimetabolous insects and holometabolous insects. The Drosophila is one of those holometabolous insects. That means it goes through a complete metamorphosis. In the first embryonic development leads to active feeding stage called larval stage, and then there is what's called a pupal stage or cocoon stage in case of silkworm terminology, and which basically metamorphoses into a new life stage called adult stage, which is morphologically very different from the larval stage. That's why it's called metamorphosis, right? And during the larval stages, there are very different tissues or sort of progenitors of adult tissues are already kept aside, and they do not participate in the larval physiology, but they contribute to the adult development. And 4 of such progenitors are two for wing and two for halter. We call them as imaginal discs in our terminology, right? So this is, for example, wing imaginal disc. Wing imaginality is not only about the wing. Already Professor Sharma has already mentioned the wing imaginalities also contribute to thorax and to the wing. In fact, all of these things I am telling you because it's known for the last 50 years of research. But the very fact that there is one common progenitor which we call wingless at that time could contribute to both thoracic region as well as its appendage, the wing blade, is again a discovery simply based on the interpretation of the phenotype of wingless that Professor R.P. Sharma mentioned in his two papers, 1973 and 1976. And he also mentioned about very interesting. I'll come to that in a minute. So the one of the Hox proteins that my lab works. My former student Pallavi was a distinguished faculty at Translational Health Science Technology Research Institution in Faridabad this year. She also contributed immensely to this work in the lab. So UBX is a Hox protein required for identification of the third thoracic segment. And if UBX is present, you have a pair of halters, right? In the absence of UBX, you get a pair of wings. And if you overexpress what is called gain of function, of UBX in the developing wing, you will get 4 halter insect, right? So basically, UBX is required to make halter. UBX does so by suppressing wing development, and UBX is sufficient to make the halter. This is a fantastic interpretation again of a few mutant alleles of UBX by Ed Lewis and others. And Professor Ashim already mentioned Ed Lewis won Nobel Prize for this seminal work. And halter is the balancing organ, and that again was mentioned. So very quickly, how these progenitors are even specified early during development where no information is generated. As this is the early stages of embryo, this stage 11, 12, 14, doesn't matter what it is, basically depending on the time from post-fertilization, post-fertilization, what are the different stages. At some stage during development, you have two parts of the two axes of the embryo is well determined. What's called anterior posterior axis, another is called dorsoventral axis. For example, in like coordinated geometry, in all field biologists, agronomists use this x and y axis. All your graphs you use it. You call it as x axis, this as y axis, and x axis is basically your anterior posterior axis. Y axis is dorsoventral axis. Now, along the anterior posterior axis, you have very different genes which are expressed, including the segment polarity genes like wingless. Wingless is expressed in the anterior half of every segment, whereas another gene called engrail is expressed in the posterior half of the gene. At the same time, there is another gene family which basically expresses along the dorsoventral axis. One of them is called DPP, or TGF beta homologue in Drosophila. And DPP is expressed in the dorsal, and less of it is expressed to the ventral side. Wingless, which is supposed to be expressed along the entire width, anterior posterior axis is expressed, but along the entire length of the dorsoventral axis is supposed to be expressed. But DPP is also inhibitor of wingless. So happened that only regions where DPP is expressed, wingless is not expressed. In the regions where DPP is expressed, wingless is not expressed. Where DPP is not expressed, of course, wingless is expressed. But there is an intersection of these two areas is the region where the progenitors of appendages are specified, whether legs or wings of the Drosophila, right? So initially, depending on how much is DPP or wingless is expressed, you have the progenitor of the one, and which basically gets specified into a larger imaginary disc called wing disc. Again, there is a distinction. There is what is called notum, which is basically the back of the insect, and actual wing proper, right? I showed you this picture. For example, go back here. This is the notum and this is the wing proper. And the notum part is the one which is specified by this part of the imaginary disc, and this is the one which is specified by the other part of the wing imaginary disc, what we call as wing pouch. So wingless is expressed in this part of the wing imaginary disc to make wing proper. In this part of the wing disc, wingless is not expressed, which will form the notum or the back of the insect. And in mutation, what happens in, which Professor Sharma already showed, there is basically called wing notum transformation. That's why they also predicted it could be a homeotic gene. Later it turned out to be it's not a homeotic gene like UBX, right? But it is more like a early specification of the developmental event, what we call a segment polarity gene. So what happens here is, if wingless is not expressed, the gene which is responsible for the notum get extended to the region where wingless is supposed to express. Otherwise, wingless inhibits the notum forming genes and activate wing forming genes. And in the absence of wingless gene, you have notum expressed and those things. That's how you get the notum duplication or wing to notum transformation, you can call it, right? I'll show you, oops, what happened to my slides now. Okay, so here is the story which basically, the driving force for my lab research for the last 30 years is these two sentences that Professor Sharma has written in 1973 December, which we are celebrating today, right? So basically he says that there are four appendages we are talking about, 2 wings and 2 halters, right? He says, now points to the fact that there is complete autonomy in wing and halter development, right? The one is, of course, the 2 wings. One wing can develop independent of the other, although they are developed in identical size and shape, and which in fact a lot of my work in the last few years, in addition to cancer work, is also understanding how do we have organs developing to exactly the same size and shape, right? Because although they are independent of each other. In fact, this actually suggests that they are independent of each other. For example, your 2 arms, all of you look at your 2 arms, they are exactly the same size and shape, right? Proportionate to your body, there is a particular size and shape, right? But they are completely independently developed. But the end of whichever the age you are in, until the development happens, they finally end up becoming exactly the same size and shape. Similarly, the 2 wings develop to the same size and shape, 2 halter development size and shape, but you can actually have 1 wing, 1 halter, or 2 wings, 2 halter kind of insects, but the growth is not affected. And because of this autonomy of appendage development, one can actually study growth control during development, right? And the second thing, he also mentioned that the autonomy of wing and halter development, but suggests that critical stages during wing and halter differentiation are controlled by similar steps, right? That's very interesting. This is even before 1978 paper of Ed Lewis's, Nobel Prize winning paper, which came in 1978 in Nature, and where he talked about some of these aspects of wing versus halter, right? And this basically is the work that I started when I started my new lab in CCMB in Hyderabad. So what we are basically looking at is, because I started much later, 1993, my research started, particularly 95, mainly major research, and 1973 this paper. So we were basically looking at what are the events, developmental events, which set the wing developmental program, and see how much of that is present or modified during halter development as a function of UBX, because the UBX is the one which makes halter. Remove UBX, you get wing in the place of halter, right? Sorry, I think you can't see much of it because of, it's not very, I think on that screen is very, those who want to look at, you can see very clearly the expression of wingless. That what is shown in the wing, red is wingless, the top is one is the wing imaginal disc, and the bottom is the halter imaginal disc. There is a slight difference in the way wingless is expressed with the wing and halter as a function of UBX. This modification is critical to make halter, and that basically, because once wingless is modified, its expression, the downstream cascading event, because it's a long distance signaling molecule, it basically changes main expression of many other genes, leading to halter development as against wing development. Anyway, I think in the interest of time, I think I could skip some of the work that we are doing, again all related to wingless. For example, we are also trying to look at how UBX regulates wing development, suppresses wing development, promotes halter development. What are the genes which it regulates, how it recognizes targets, is a transcription factor in the, in the genome and so forth. And then we have basically been able to do, using more modern molecular biology approaches like very different gene regulatory pathways, looking at its expression using new tools. And then we have also overexpression, loss of function genes, transcriptomics, and what's called chromatin immunoprecipitation methods and so forth. And amongst the many genes that we have identified that which are targets of UBX, which are very critical for Halter specification, is mostly the wingless is the primary one. And because we could start working on wing and halter differences as a function of UBX, and downstream of its wingless is regulated, we could also use the wing and halter development as a genetic assay system, right, to identify new components of the system. For example, we have discovered that there is a particular gene called twins. It's basically a protein phosphatase 2A family member, and it's a positive regulator of wingless. And this basically, you can, as a positive regulator of wingless, downstream of wingless, when you have a loss of function mutation of it, it should actually mimic wingless mutation, right? For example, is exactly what happens. We do have wingless mutation or wingless phenotype when this particular gene is mutated, and you also see no-term duplication, exactly like what wingless does, right? Now, we also have looking at the more molecular where UBX regulates downstream targets. I'll not get into the details of so much. We also do comparison across different insects. This is more of an evolutionary developmental biology kind of a program. For example, Jubilee Drosophila have 2 wings and 2 halters. All other insects have 4 appendage, flight appendage, insects like Lepidopteran, moths and butterflies, Hymenopterans like bees, and Odonata like dragonflies. Coleopterans also have 4 major appendages, but the front 2 are the modified into elytra, the thick covering ones you see in bumblebees. And we have been able to compare using very different genetic methods, what may happen, and we basically ended up saying that there is the ability of UBX in Drosophila to downregulate wingless signaling is only present in Drosophila and not in other insects. And then we started looking at what could be the reason why wingless signaling is downregulated in Drosophila but not in other insects, which is basically critical for developing halter in Drosophila, and whereas wing is developed in, let's say, Lepidopteran or in Hymenopterans like honeybees. I'll not get into the details, it's already published, it's in the papers. I'll just quickly move on to the cancer part of my work because we are already late. Okay, all of you may have heard about colon cancer, right? Professor R.P. Sharma talked about tissue regeneration. We have what's called stem cells, and these stem cells are basically division competent cells, because once cells differentiate, unlike in plants where cells can de-differentiate and become cell division competent, in animal tissues, once they differentiate like muscles, neurons, epithelial cells, whatever they are, they cannot divide again. They completely come out of the mitotic division phase. But there are certain cells which are kept aside as stem cells which are division competent, but they divide only when there is an external signal, and that signal could be dying cells or because of injury, because of simply wear and tear. Sometimes it could be simply programmed. For example, inner lining of the intestine, it is replaced once in every 2 weeks. Every cell in the inner lining is replaced by every 2 weeks, and it is completely programmed. And Wnt signaling is the one which is involved in replacing the inner lining of this one, because in all kinds of toxic things that we eat, the pH is also very, very low in our stomach, intestine. So inner lining has to be replaced much more quickly than, for example, cardiac cells or liver cells or lung cells, right? So the people have noticed that there is what is called colon cancer, and that colon cancer that is associated with a mutation in a gene called APC. Much before wingless pathway was discovered or elucidated, APC mutation was already known. People knew that people have a mutation in APC have high risk for colon cancer. And again, cancer is an emergent property of having stem cells in the body for regeneration. If there are no stem cells, if there is no regeneration, at the same time there is no cancer. So cancer is the cause, price that we are paying to have the regeneration capability. Without regeneration, we can't even survive. None of our organs will survive without regeneration capability. So in a way, it's a basically in evolution, there is always something, a cost to everything that has a benefit, right? So similarly, cancer is a cost or the price that we pay to having the benefit, having this regeneration capability in our body. Anyway, colon cancer is one such cancer which is associated with the mutation in APC, and people later, and when Wingless signaling in Drosophila and Wind signaling in mammals were elucidated, it turned out to be that APC is more like a scaffold protein, which is a very large protein, 3000 kilodalton protein, which is basically made, the CDN itself is close to about 10 kilobase pair, right? Or mRNA is 10 kilobase pair. So when there is, Wnt is basically a signaling molecule, it's a long range signaling molecule. It's a ligand which basically comes outside the cell and sort of goes through the extracellular matrix and binds to a receptor called frizzled. And when there is no Wnt signaling, what happens is there is a particular protein called beta catenin. Again, Professor Sharma has alluded about beta catenin. Beta catenin is basically bound by APC. Once it's bound by APC, there is another kinase called GSK3 beta which phosphorylates beta catenin, and this phosphorylated as a beta catenin is like a label that it should go into degradation pathway, the proteasomal degradation pathway. So there is no beta catenin in the cytoplasm or in the nucleus when there is no Wingless signaling. When Wingless signaling is present, it activates the receptor. When the receptor is activated, the APC beta catenin, that complex, get dissociated because there is no phosphorylation of beta catenin. In the absence of phosphorylated beta catenin, APC get dissociated. Now beta catenin go to the nucleus and turn on so-called Wind target genes or Wingless target genes, which basically could be related to differentiation, proliferation, and so forth. In the context of stem cells, it's of course to induce proliferation. Now, in the absence of APC, what happens is, there is constitutive expression of beta catenin, leading to continuous proliferation of these epithelial cells and formation of tumor, and tumor will eventually lead to become the cancer disease. And what we did early during our time in CCMB in Hyderabad is actually use Drosophila. One of the advantages of using Drosophila is not only writing Drosophila development or studying Drosophila, molecular biology and trying to correlate to, a vertebrate or mammalian or human, biology. You can actually use Drosophila as a model to study human genes and human diseases. So this particular work basically was, because it's a 3000 amino acid long protein, we wanted to basically look at what is the genotype phenotype correlation, the mechanism of genotype phenotype correlation in case of APC using Drosophila as a model, because there are so many different ways. There are mutations which are known in human, but not all of them will lead to cancer. Some of them will lead to cancer, some of them will not lead to cancer. Some of them are highly dominant and lead to large, higher severity of cancer, and some kind of is a minor or a less frequent version of the cancer. So we basically looked at all kinds of combinations of these, because you can make transgenic Drosophila very easily. You can express human APC of very different versions, wild type, mutant, and very different deletion versions under different contexts in Drosophila development. And then basically we try to establish that some mutations basically have dominant negative interactions, some of them have additive interactions. That's one of the reasons why you can get variation in the phenotype. We also use Drosophila as a model system to look for anti-Wnt inhibitors or anti-Wingless inhibitors, and then you can use actually some kind of a drug screening method, using Drosophila eye development as an assay system. Again, it doesn't matter where Wnt signaling is required, you can use that to study Wnt pathway or Wingless pathway, and at the natural phenomena. We can also use that as an assay system. Particularly eye development is easy for assay system because it's easy to score under microscope. Okay, I think I'll stop here. It's been a fantastic journey for all of us, and fly biologist, particularly personally for me, having worked on wing development, heart rate development, wingless signaling, wind signaling, and human cancer. For me, Professor R.P. Sharma and his work has been extremely important. And also I had a very good personal relationship with R.P. Sharma. In fact, when I was awarded Bhatnagar Prize, he was the first one to invite me to give a lecture in IRA in genetics department, and it was a privilege to be honored by him. And Professor V.L. Chopra, I don't know, for some reason he liked me so much. Unfortunately, he's not there. I was like early 40s, and he was such a senior figure, a former director, planning commission member, and everything. And he started talking to me regularly, and he invited me for meetings, and he made me a committee. In fact, a very high-profile committee of ICAR to decide all very different awards and some kind of a reforms of ICAR awards. So there was only a 3-member committee. V.L. Chopra was the chair, T. Ramaswamy at that time secretary DST, and myself. And the 3-member committee was established, and V.L. Chopra always gave me some space, not only in science and also in science administration and policy. And I'm really indebted to both of you. Thank you very much.
Thank you, sir, for such an enlightening lecture.
On behalf of the fly community, I want to present this to Professor Sharma.
May I now request Professor R.B. Singh, Chairman of today's event, for your valuable remarks.
Oh. Friends, it's a very difficult job for me to really summarize the wonderful presentations which have been made, or to give you some guidelines from that. But let me try to see if some message has emerged which possibly may help all of us, particularly our aspiring students and young scientists. A message is very clear. Before I proceed, I wish to doubly congratulate Dr. R.P. Sharma for the wonderful work which is so stimulatory that a large number of people have benefited all over the world, including some even winning the Nobel Prize. Professor Sharma, congratulations and congratulations. You and your team. And this only tells us that while you are doing some research, please concentrate with your full devotion. And even if you fail for first time, second time, third time, don't be disappointed. With devotion, success will be yours. I am sure you will succeed. This is the very first lesson that we should be able to get from these presentations. Two, I see here that there is a need for really conjecturing not only, but looking into the reality of the connectivity of the basic research, strategic research, applied research, and back and forth, each of them enriching each other. So this is very important for us to keep the continuity right from the very fundamental basic research up to the applied research, curing the cancer from the level of the knowing the gene sequence of the wingless mutant. They are all interrelated. So you can see here the connectivity, the continuity of the basic, strategic and applied research. And to allow that to happen, we need more Swaminathans, the visionary outlook, the vision. In IARI, it's not only rice and wheat, it is not in livestock or this or that in the agricultural institutions, but particularly if you have to really do something depending on the best of the science to begin with. He took lesson from Yokohama, and you can radiation genetics and mutation breeding, all that combination with the physical and chemical mutagens combination, how to moderate, how to increase the efficiency, how to give direction to mutations, and how to really make use of these. That kind of, and establishing a Drosophila lab at IARI, it's all a matter of vision. So this is the other thing which I found, that one has to be visionary, one has to be Swaminathan to really see that there are ways by which unusual things, this is called disruptive pathway. That disruptive pathways you adopt, you will reach somewhere which you could never imagine. Disruptive pathway. Historically, the things the way were narrated by Lakhotia Sahab, Professor, amazing. We all agree, Drosophila is not the queen only, it's Cinderella. And how much it can really do and contribute to it. And I am so pleased that both at IRI and BHU, that we see here what difference it can make. The scientists, what I thought was very important, that the scientists can go to the Prime Minister, and the Prime Minister will listen to the scientists to make a change in the contribution or enriching the facility at a particular place to enhance science. I have been always telling, why our scientific community, the meeting that we are having, Ashok yesterday and today that we will be continuing, how many times we mentioned, let scientists tell the truth and let scientists go to the Prime Minister. If something is required, I am sure science seeks truth, and that truth is going to prevail through your real discussion with the top man in the country. You will get that support so that you are able to carry on the right science. And I am so glad Lakhotia Sahib mentioned that how it made the difference. This is really wonderful, and I am very pleased that we will be able to take it into consideration. I am not going into the scientific part of doses compensation and sex determination mechanisms, whether it is the XA system as you have in Drosophila or the XY system in the human being. These are all matter of doses compensation, gene action regulation. And this is how I would like to connect that how one discovery can result into, and this is how you can also see, if you have one system, you will allow, if the XA system of sex determination is there, you will follow one pathway. If the XY system of the gene control and the sex determination is there, you may also have polyploidy in the system, which otherwise XA system does not allow polyploidy, the pathways. So your power of thinking You have to deviate your power of thinking to really come up with some new ideas, new way of doing things which you can apply. And this is where Ayurvedic medicines, and if Ayurvedic plants have to be changed, how probably you can see the utility of some of those discoveries, and the stress-induced genetic modification which leads to epigenetics. If a stress can lead to genetic modification, it comes to epigenetics. And you know, our children in India, the world's 1/4, 1/3 of the stunted, wasted children are Indian children. They have their homes here. If that is stress of undernutrition, stunting or wasting, can bring a genetic change, as we have heard. Similarly, that epigenetic effect should be able to tell us that we please manage yourself. Don't allow the stresses, don't allow this high percentage of undernutrition and stress to the children. If your child is undernourished, stressed, the intellectual development, the brain development and so on affected, where will we go to? Your country is depressed automatically with that high degree of stress impacted epigenetic effect. You are all seeing this thing happening. So this is another lesson that we learned from the presentations and the discovery. Of course, we knew that the foresight, observations of structures and facts, relationships, the incremental science, the word which I think is so very important, the incremental science that we have to go on pursuing will be based on your understanding of each step by step as we go along. Only then you can get the incremental gain, not jumping, not simple jumping. Therefore, the regulatory pathways, the way that we were able to learn, the differentiated developments which lead to the evolution and the various kinds of differentiations. We also see here that the insight, if you look into the major sufferings that human beings are through one or the other kind of disease, including colon cancer and so on and so forth, how they are interrelated with the stresses and also with the gene mutation or with the deficit or the loss of a gene. So I see here that various interconnectivity, and I would like to congratulate the two outstanding speakers, Professor Sharma, your own original presentations that you gave, and the connectivity and the enthusiasm that it should be able to build. I will request the Director of the Institute, Honorable Dr. A.K. Singh ji, please. I think I never knew that here is such a gold mine, the Drosophila lab. I think if some of the young generation students want to do research on the beginning on some of those aspects of Drosophila, allow them to do this thing here. I think it is a knowledge center, and that will be again very helpful. I would be happy to see this complete rejuvenation, strengthening of the Drosophila in the Research Advisory Committee also. Let us make a recommendation to this effect, that let that be a program, it should be strengthened. So with these words, my friends, I want to again, Professor Sharma, congratulations, the two outstanding speakers supporting the way that we should proceed. All the support the director has provided to this genetics club, Gopal, and all my distinguished colleagues who are here listening, and more and foremost, these dear students. Students, you are the future of teachers tomorrow. You are the future Nobel laureates. You are future R.P. Sharma. Please take lessons from that. And I wish you all the very best in the years to come. Wish you all the very best. Thank you. Thank you.
Thank you, sir, for your valuable insights and providing us with the take-home messages from the event. So, ladies and gentlemen, as we draw towards the conclusion of this remarkable event, it is our distinct honor to express our deepest gratitude to our eminent guests who have enriched us with their presence and insights. May I now request our beloved director, Dr. A.K. Singh, to kindly do the honor of felicitating our chairman of the session, Professor R.B. Singh, with a shawl and a memento as a token of our love and respect. So, before felicitating Professor R.P. Sharma, we have a citation prepared on him. With the permission of the chair, I would read the citation for you. Dr. R.P. Sharma, a distinguished scientist and geneticist, has left an indelible mark on the landscape of developmental biology during 1973 with his groundbreaking discovery of a wingless mutation in Drosophila melanogaster during his tenure at the Indian Agricultural Research Institute. Born on 20th February 1940 in Roorkee, Uttar Pradesh, India, Dr. Sharma's illustrious career has centered around unraveling the mysteries of genetic pathways that orchestrate embryonic development. His pivotal contribution was first published in December 1973 in Drosophila Information Service and later as a comprehensive research article in Developmental Biology co-authored with Professor V.L. Chopra. The wingless gene, aptly named for its role in adult wing formation in Drosophila, became the cornerstone for understanding intricate developmental genetic mechanisms not only in Drosophila but across the animal kingdom, where it was known as INT1. Identification of WG1 as an orthologue of INT1 gene led to the coinage of a new nomenclature, WINT1, amalgamating both the names. Beyond the confines of the laboratory, Dr. Sharma's discovery proved to be a key that unlocked our understanding of developmental defects and their links to human cancers, including colorectal cancer and skin cancers. The WINT1 pathway, deciphered through systematic genetic approaches and mutation analysis, continues to be the focal point of developmental biology research even after 4 decades. Acknowledged for his outstanding contributions, Dr. Sharma has been honored with awards such as Om Prakash Bashin Award, Science Congress Millennium Plaque of Honor, and the INSA Silver Jubilee Medal. His fellowship in prestigious institutions including the Indian National Science Academy and the National Academy of Sciences, India, underscores the significance of his work. At this Golden Jubilee year of the landmark discovery of wingless gene by Dr. Sharma, IRA recognizes its monumental importance that has not only shaped the trajectory of developmental biology, but has also paved the way for understanding the complex landscape of wind signaling in both model organisms and human health. Thank you. May I now request the dignitaries to kindly do the honor of felicitating Professor R.P. Sharma with a shawl and memento. May I request the dignitaries to kindly do the honor of felicitating the chairman of the session, Professor R.B. Singh, with a shawl and memento as a token of love and respect. I also request the dignitaries to kindly do the felicitation to the keynote speakers of today's event, to Professor S.C. Lakotia, and to Dr. Professor Shashi Dara, sir. Thank you, sir. Your presence has made today's event truly memorable, and we extend our heartfelt thanks for sharing your time and knowledge with us. So, as we wrap up the event,
we can see the former faculties from the Division of Genetics, and also project director, director of sister institutes, heads, professors, students, ladies and gentlemen. It is my proud privilege to present the vote of thanks. On behalf of the Genetics Club, the Genetics Congress Trust, and the ISGPB, and the entire genetics fraternity, and on my own behalf, extend a very hearty thanks to Professor R.P. Sharma for kindly accepting our invitation for the. I would like to thank Dr. Mohapatra, our guest of honor, for initiating the thought process of celebrating this event. Essential remarks at the beginning of the event. Professor S.C. Lakotia, the distinguished professor from BHU, for enlightening us on the fly research in India, of the pedigrees and lineages, and also how he has connected efficiently from fly to pharma in the form of Ayurvedic medicine in preventing neurodegeneration. And I would like to place on record our heartfelt thanks to Professor L.S. Sashidra, Director, NCBS, Bangalore, for his excellent talk, as well as accepting our invitation for presenting his talk, and also explaining that science is incremental, and also enlightening us that breakthrough discoveries always provides important clues for follow-up research. I would like to extend our sincere thanks to Professor R.B. Singh, who is an ocean of knowledge and who is always with us for gracing the occasion and also chairing it, and also emphasizing the role of devotion in research, continuum in research, visionary outlook in research, disruption in research, and also parallels in research, and the power of thinking in research. Thank you very much, sir, for your presence and also your chairman's remark. I thank our director and vice chancellor, Dr. A.K. Singh, all the joint directors as well as directors of sister institutes, project director and members of Genetics Club for making this program a great success. I see former faculty members who have been kind enough to be present. Thank you very much for your kind presence. And also some key members of the organizing committee I would like to name at the cost of missing some. Dr. Akshay Talukdar, Dr. K.K. Vinod, Dr. Harita, Dr. Chandan, and Dr. Ramaprasad. I would like to end with a couplet number 3 to 4 from Tirukural on virtues. I quote, "Opa talaigudi ulla peridal anaithe pulavar tolil," which roughly translates as it is the prowess of scholars that meetings bring delight and departures leave memories. I wish that this Golden Jubilee commemoration of the discovery of Wingless, which we witnessed today, brought delight to all of you and will be etched in our memory for times to come. Thank you very much once again.
Thank you, sir. Thank you all for joining us and gracing this occasion. I request you all to join for a cup of tea arranged outside this auditorium. Thank you.